microorganism we have with us is the algae. Now, on a very heavy rainy day during the rainy season, over the while you see a very slippery growth and on the floor, and that growth is green in color actually. So that is nothing but that is the algae. So algae is usually found on wet and damp places, and since it's found in the wet and damp places, and its structure is designed in such a way that it contains chlorophyll. Now we know that in this structure right over here, that's the structure of the algae. This is the algae structure. So in this structure, what you find is it's present in form of filaments, and the growth you see is why so widespread and so slippery, because of this structure. So now the next point we learned was that it contains chlorophyll. In this whole structure, what you see is these white dots. These represent the pigments that would have chlorophyll in them. So since we know that chlorophyll over here is a pigment that is responsible for performing the activity of photosynthesis, and we know that by the process of photosynthesis, a plant can produce a food for itself. So the plant which is able to perform photosynthesis by the help of chlorophyll are known as autotrophic plants. These autotrophic plants are the plants that I just now told you can produce food for themselves. So how will you define autotrophic plants? Autotrophic plants are the plants that are capable of producing food for themselves by the process of photosynthesis. So I will just write down photosynthesis over here, because that is the prime source by which the plants perform the activity of making food for themselves and releasing oxygen in the air. But we also know that algae are useful, and some of these are useful to serve as the food that we consume over here, or the plant animals that consume the food, the green leafy things. So we also know by the algae being autotrophic, we know that we are dependent on it as the food source, and so are the herbivore animals. So algae is useful because it produces some kind of algae. They produce food for the herbivore animals. That is why they are also of importance to the mankind. The next point we learned is that if they grow in an overhead in our tank, and that tank is filled with water in it, so what will happen is there would be a very foul smell coming up from it, and the whole tank would be having a foulish texture to all of it, to the surrounding of the tank, and especially when it gets exposed to the sunlight, the water is going to smell very off, actually very rancid in nature. So why is this? Obviously because the tank is having water in it, and the water is stored for many days inside it. So whenever you actually just observe this by trying it at your place once, take a vessel that is made up of plastic or any other material and fill in water in it and leave that water for 2 days. After 2 days, you'd find a very slippery surrounding to the sides of the water. So this would be an indication that there is a growth of algae. So similarly in tanks, when there is a lot of water stored inside and there is direct sunlight being exposed to it, there are chances that in this tank the algae is grown on the sides or on the bottom of the tank where the water is exposed, and finally it would result in giving a very off smell to the water and a very stagnant texture to the whole of the tank, and it would not obviously be good because this is not a healthy sign actually for consumption. So that is why this process, it should be avoided by regular maintenance of the tanks, by adding bleached water, by keeping bleached water so that these processes of algae growing is slowed down and make the water more potable for us. The next point is that the diatoms are kind of a unicellular algae over here. So this is just one structure that is a unicellular algae, and this is a diatom. So this is nothing but a unicellular algae. Now we'll just repeat the whole answer once again. So what we learned in this was that algae are found in wet and damp places. We also learned that these algae structures, they contain chlorophyll in them, and we know that chlorophyll is one pigment that is responsible to carry out the whole procedure of photosynthesis. Now a plant can produce photosynthesis. That kind of a microorganism or a plant can be known to be autotrophic, because it produces food for itself. It does not depend on any other source or any other medium for the need of food. We learned that it is also useful for because certain type of algae, they produce food, and this food is usually consumed by certain types of herbivore animals. So the next point we learned was, since everything has a good side to it, there are certain aspects that have to be bad to it too. So the point that we learned in the bad aspects would be that if they grow in an overhead in tanks, the tanks that has water in it actually, and when they get exposed to sunlight, they would create a very bad smell or an off smell. Next we learned was that the diatoms are kind of the unicellular algae. Over here, this algae structure, we learned that these are the diatoms of algae, and this is one unicellular structure of the algae. Now let's move on further to understand a little bit more about fungus. So right here I have the structure of fungus with me. Now this structure is, as you can see, it's filamentous. It has a hyphae and it has extended spores. So when this bursts, this spore, a lot of spores are released in the air, which finally go and propagate in various media when it is in like wet and damp regions. So the structure we've studied, it was very basic. It had hyphae, it was filamentous, and it had spores in it for the purpose of its propagation. Now we learn a little bit more about the fungus. So in fungus, some funguses are useful, while some funguses are not really useful. Like these funguses either can cause disease on your skin, or they can damage the substances that they are exposed to. So why is this all happening? Because these funguses, they do not have chlorophyll in them. That means no chlorophyll, so no photosynthetic process, no food material that is produced, no food material, so no energy that is acquired from them. So right here is I have the structure of the fungus that is drawn on the board. Now you can observe something in this structure is that the structure has long filaments and it has hyphae extended out. So we can brief it out with the structure that the structure of fungus is filamentous, has hyphae, and over here, right here, you see these little spores. When this mold is bursted, these spores, they are released into the air and they are propagated to various spaces, to various damp and moist spaces where they can have a very reasonable environment for growing and multiplying further. So let us just learn a little bit more about the fungus. Now in fungus, we know that there are some funguses that are useful, while there are some funguses that are not useful. So the funguses that are not useful would result in causing skin diseases or damaging the substances that they're exposed to. Now, why would that be, that when they damage the substances that they're exposed to? Because that is because they have no chlorophyll in their whole body structure. This body structure, this whole cell structure actually is not having chlorophyll in it. Now, because of this structure, the microorganism structure, and no chlorophyll present in it, that means that the process of photosynthesis would not be performed well, and because of this absence of photosynthesis, the microorganism, that is the fungus, will not be able to retain energy, because there's no food made from this process of photosynthesis, and this would finally lead to its dependency on another organic substances for the mode of nutrition to be acquired and sustaining its life. So that is known as the parasite, right over here. Now, parasites may be defined as any living organism or any microorganism that is dependent onto another organism for filling its needs of nutrition, either shelter or any other demand of the living organisms that need to sustain their life. That would be known as the parasites. The next one is that they grow on damp places on organic substances. Now, since I told you that these are parasites and it depend upon the other materials, other organic materials, for the mode of nutrition to be completed or for having any other basic necessity of them, they need any other substance to grow on, and that is nothing but the organic substances, because only organic substances that are present in damp places and would have a perfect content of moisture and perfect level of nutrition that would be giving this structure of fungus would be fulfilled only by the organic substances. So that is why these are the parasites, because parasites would obviously be dependent upon the organic substances only to fulfill their needs. Inorganic substances would not help them in any way. So the next one is that not all fungus are actually harmful. There are certain funguses that are helpful also. So there is this one fungus named Penicillium, which is used to produce or create an antibiotic known as the penicillin. Now, this discovery was made by Alexander Fleming, and he was just growing a culture and he was trying to perform some other experiment, but at that time only there was an outbreak of these diseases which demanded certain antibiotics for the cure. So it was a discovery at that time that he could recognize that this penicillium, that is the fungus, could have the ability to produce an antibiotic that is known as a penicillin. Now, antibiotic is nothing but a substance that can kill certain types of microorganisms over here. Now, these certain type of microorganisms need to be killed because they are harmful to the whole living environment, living organism like animals, plants, or even the human beings for that matter. So now about the point about producing antibiotics. Not every fungus is able to produce antibiotics because only a few microorganisms can only produce the antibiotics, and it is not necessary that only fungus can produce antibiotics. There are other microorganisms too that produce antibiotics, and these antibiotics are useful for killing microorganisms that create diseases for the other situations and other problems. So this is the whole brief about fungus. But before moving on to the next, that is the yeast cells, we will be learning a little bit more about fungus. I will be explaining to you about the table actually for the antibiotics that is required, that is actually necessary to learn, because you need to learn about the basic antibiotics and their usage in the types of diseases and their importance in helping the whole mankind. But before that, we would learn about this in a little more detail. We'll learn about it once again. So I explained to you about fungus, about its structure. Now what I explained was that this is the structure of the fungus, and as you can see over here, the structure is filamentous, has hyphae, has spores for its propagation. So it's very basic and very simple. Let's move on to the next part. Now in fungus, there are some funguses that are useful, while there are certain funguses that are not useful, and they are not actually helpful at all. So these funguses that are not useful would be the ones that cause skin diseases or damage to the substance. Now since they have no chlorophyll in it, they cannot perform the activity of photosynthesis, and they are not known as autotrophs, obviously. So since they cannot produce this process of photosynthesis, they have to be dependent on any other medium to fulfill and replenish their needs of nutrition and shelter and other basic needs for them. So basic needs would include the moisture content and all the other factors. So they are known as parasites actually, and these parasites, they grow on damp places on organic substances. As I told you, moisture content is really important for fungus growth, and the organic substances are important for the fungus's nutritional content. So this is important for fungus's nutrition. Now, since I told you in the start right over here that there are some funguses that are useful too. So one of them is the Penicillin fungus that produces an antibiotic named as Penicillin. And only a certain microorganisms can produce certain antibiotics. Not all are capable of producing antibiotics. And antibiotics are those substances that can kill certain type of microorganisms. Certain type of microorganisms that cause illnesses and diseases and outbreaks.
No more spending your energy and money on coaching classes, or missing classes for rain, for rally, and any other nonsense. Subjects covered by multiple teachers with repeat telecasts. Doubts, concepts, applications, no problem. All explained through great and significant animation. So sit back comfortably in your home and watch Study Spectrum TV channel.
So I explained to you that there are some microorganisms which are able to produce antibiotics. Not all are able to produce these antibiotics. So some of these are the organisms that have the capability to produce antibiotics. Now just let me explain you what antibiotics again is. I told you previously that antibiotics are the substances that are useful in killing the microorganisms that are harmful to the body. So I have a table over here that consists of the antibiotic product that is being created by the microorganism and the type of disease or the type of infection it is useful in curing. So the first table is antibiotics produced, the second one is microbes producing the antibiotics, and the third one is the germs destroyed. Germs is nothing but the pathogens that are actually present in this. So the first antibiotic is the penicillin, and the microbes that are useful for creating this antibiotic is the Penicillium chrysogenum, and the disease it is useful in curing or treating is the diphtheria, or rather the pneumonia pathogens. Now I told you that Penicillium chrysogenum is a fungus. So let's move to the next one, that is the chloromycetin. Now chloromycetin is produced by the microorganism that is Streptomyces venezuelae, and Streptomyces venezuelae is responsible to cure the typhoid germs. Then we have along with us the streptomycin. The antibiotic that is present is streptomycin, and the microorganism that produces this is the Streptomyces griseus that cures tuberculosis germs. Now many a times when these patients they get tuberculosis, and it is very contagious because it is an airborne disease, it spreads through air and it is so easy to contract by any healthy host also. So there are doctors who ask preventive measures, but only preventive measures are not responsible for actually curing disease like tuberculosis. So what do the doctors suggest is a antibiotic. Now these antibiotics, they go and they act on the pathogens and the germs, and they kill them, and they sustain and they stop and cease their activity, hence by curing the disease. So the last one we have is tetracycline. Now tetracycline is a very widely used antibiotic, and it is produced by Streptomyces aurefaciens, and it is useful in treating various kind of germs. So in the field of biotechnology, actually when experiments are conducted, it is of prime importance in biotechnology that there are no microorganisms that are harmful for the growth of the other cultures to take place actually. So this is one prime and one important antibiotic that is useful in making the cultures. So I'll just repeat this table again for you to go thorough with the names. So the first one we had was penicillin, was the antibiotic that was produced by Penicillium chrysogenum. Penicillium chrysogenum to cure the disease of diphtheria. Then the second one we have is the chloromycetin. Chloromycetin is made by the microorganism Streptomyces venezuelae, used to treat the typhoid germs. Then we have streptomycin. Streptomycin is produced by the microorganism Streptomyces griseus and is useful in treating tuberculosis. Then we have the last antibiotic, that is tetracycline, and the microorganism that produces tetracycline is Streptomyces aurefaciens, and this is used to treat various germs. Now when you are ill with any of these diseases or germs that have infected your body, there are doctors who will prescribe you these 4 antibiotics very often. So these are the antibiotics that have actually revolutionized the medical field and are proving to be really helpful and prevent the outbreaks of diseases. So let's start with the last 2 types of microorganisms, that are the yeast cells and the protozoans. Now in yeast cells, we know that they belong to the class of fungi, and hence they have no chloroplast in it, and they are not autotrophs. Actually they depend on the other sources for their nutritional requirements, and these yeast cells are actually the main cause of fermentation. Now fermentation caused by some microbes results in the formation of organic substances with excellent taste and flavor, and these may involve like breads, idlis and dosas. So while there are certain fermentations which are actually not desirable, and these non-desirable fermentations will lead to the production of very unwanted odor and undesirable texture of food, and this food is actually not at all worth consuming, and it's not healthy at all. So as I told you that every side has a good and a bad part to it. So let's move further with the protozoans now. Now protozoans belong to the unicellular animals. That means they have just one cell in their body, that is, they are unicellular. They do not contain chlorophyll in them, and the cell membrane and nucleus are the only organelles that are present. So they are unicellular, they have no chlorophyll in them, and their body consists of only the cell membrane and just a nucleus in it. So one example of this protozoa could be amoeba. So amoeba, we all were introduced in a very younger standard that it had no shape to it, no size to it. It was just like a pseudopodia, could take any shape, and pseudopodia is the false feet. So that is one protozoan, and protozoas were the very first originated microbes on the planet. So then there are not all the protozoas who are actually useful and helpful. There are certain protozoas which are harmful. So these protozoas that are harmful are like the amoebas. When these protozoas are introduced in the food, or they take up the nutrition from food that you are having, and they enter your body, they are more likely to create infection into your body and give you a tough time recovering to it. They may give you infections like diarrhea and vomiting, and infect your intestine and your whole of the body system. And if this is not cured and not treated on the right time, this would lead to worse situations where you might have chronic effect of it on your body. Therefore, while you observe any changes in your body that are not usually seen, it is most likely to go and visit a doctor and cure them. So let's just repeat these two once again. So yeast cells, they belong to the class of fungi. They cause fermentation, and fermentation is caused by certain microorganisms like the yeast cells, to be precise actually. And these changes, the fermentation results in the change of formation of organic substances that has excellent taste and flavor. So while there are certain fermentations that are not desirable and they lead to odor and not a very desired texture of food, and these are not worth consuming at all. So the examples of fermentation that are consumable and have a desired quality and texture of food are bread, idlis and dosas. So the next one we go is to protozoas. Now protozoas, they belong to the unicellular animals. Unicellular animals means have just one cell, and they do not contain chlorophyll in them. Instead, they just contain one nucleus, and that is surrounded by the cell membrane. So there are certain protozoas that are harmful, just like amoeba. Amoeba can cause diarrhea and vomiting once it enters your intestine and infects your body. Since it does not contain chlorophyll, it is dependent upon any other source for its mode of nutrition. So here we are done with the types of microorganisms, studying about their structure, studying about their functions, studying about how they can affect human beings or animals or plants for that matter. So now we'll just have a brief summary about what we learned right from the start till the end. So what we learned was that these cells are the fundamental units of life, and they're the building blocks of life. Without them, the whole life processes wouldn't be as easy and as possible as it is now. So Robert Hooke was the first scientist, the first American scientist or the English scientist that discovered in 1665 under a microscope. He observed a section of cork, and he observed that it had compartments in it just like a honeycomb maze had. And he named this to be as cells, and from there the advent and the introduction of the word cells came in the picture. We also learned that the study of cells is known as cell biology, while the study of cytoplasm is known as cytology. Then we learned about the microscopes. We learned about a compound microscope, we learned about an electron microscope. Now we learned that in a compound microscope we could observe things that we couldn't observe from our naked eyes, but electron microscope had around 2 billion times more magnification than that of a normal compound microscope, and it could observe the organelles present inside the cell. With the help of compound microscope, we could only observe the cell structure but not the organelles and the components inside of it. But with the help of electron microscope, we could study that, because electron microscope had a magnification power of more than 2 billion times than that of a compound microscope. Now we also learned that the cells in a cell, there are these components, constituents that are embedded in the cell, and these are known as the organelles. Then we went on to study about different type of organelles. We learned about the plant structure and about the animal structure of cell. In both, we found that the organelles were common, but in plant cell there were two certain additions to organelles. Now we'll discuss about the common organelles that were present in both plants and animals. We found that the plant and animal cells had nucleus common to it, they had the vacuoles common, they had Golgi bodies, they had mitochondria common in it, and they had cell membrane and cytoplasm in common. So we'll start with the cell membrane. Now cell membrane is the outermost covering of the cell, both in plants and animals. So in plants it's the innermost covering, and in animals it is the outermost covering. So we learned that in plant and in animal cells they had one membrane common, that was the cell membrane. Now cell membrane is nothing but it is very flexible, it is very thin, and it enclosed all the organelles inside it in the cytoplasm. Then we learned that it performed the main functions of protecting the cell and giving it support and attaching cells together. Then we learned that in plants the cell membrane was not the outer covering only. It also had the cell wall. Now cell wall was a very rigid covering made up of cellulose